Hi friends, welcome to Free Life Science class. We are going to discuss about basics of DNA replication. So in this video, we will only handle the basics of DNA replication, the key steps of DNA replication, what are the factors required for DNA replication, what are the enzymes required for the DNA replication. All that things will be discussed in this video. That is, this is the part 1 of DNA replication, and a further mechanism will be explained in the part 2. Okay, so we can move into the basics of DNA replication. Okay, so you know that DNA replication is the process of making a new copy of that DNA itself. So your cell divide 3 billion, your cell divides every day. So there are 3 billion base pairs of DNA in your genome. It should have to copy it accurately so that the new daughter cell will get same copies of the DNA, isn't it? So this is a cell. It consists of 3 billions of genome, base pairs of genome, and exact copy of this DNA should be copied to the new cell when a cell divides, isn't it? So the DNA replication is very important process of existence of an organism, so that we can say DNA replication ensures that an exact copy of DNA is passed to the daughter cell during cell division. Okay, so if your cell fail to move to divide, or it cannot replicate, then what will happen? This process, that is cell division, will not take place. It will arrest in the G0 phase. You know G0 phase. You know the cell phases, cell cycle phases, that is interphase and mitotic phase. In interphase you can see G1, S phase and G2 phase, and in the mitotic phase, that is M phase. So this G1, we can say it is a preparatory phase. If your cell fails to do something, or if your genome fails to make some factors for DNA replication in this phase, then it will not allow to this phase. It will not allow to this phase. Instead, it will go for a resting phase, an arrest phase, that is G0 phase. At the same time, this is the synthesis phase, S phase. So here your DNA is replicating. In this process as well, if your DNA fails to replicate, it will move to G0 phase. Okay, then only S phase is transferred to G2 phase. Okay, so if your cell failed, what will happen? What will happen? If your cell failed for replication, failed for replicating the DNA, what will happen? The cell moved to a G0 phase. That is, we can say mitosis or meiosis will come to halt because the cell failed to replicate. Okay, now we can move into DNA. DNA consists of 4 bases: adenine, thymine, cytosine and guanine. If you are considering RNA, instead of thymine we can say uracil. Okay, so the 4 different bases in DNA are adenine, thymine, cytosine and guanine. Beside these 4, any of other bases like uracil or hypoxanthine, ribothymine, etc., like that is present in the DNA, it is an indication of mutation. So there should be no unusual bases present on the DNA. So the bases present on the DNA are adenine, thymine, cytosine and guanine. Okay, The adenine always pair with thymine and cytosine always pair with guanine. There are two hydrogen bond formation between adenine and thymine, and there will be three in between cytosine and guanine. And the backbone of DNA is made up of bases. Definitely we have discussed about bases. So it consists of bases. Then about sugar. So DOC sugars are present in the backbone of DNA. Sugar is present. Then about phosphate. So phosphate is also present in the backbone of DNA. So these three are known as nucleotide. These three together is known as nucleotide. Okay. So what is the backbone of DNA? The DNA is made up of bases, sugar and phosphate. Okay. Next we can move into a mechanism of DNA replication. I am going to say the DNA replication in 4 steps. That is, first of all, unwinding of DNA. So our DNA is in the form of double helix. So it should be unwinded. Isn't it? Our DNA is in the form of double helix. So we have to unwind it. Then only the replication will takes place. So then the hydrogen bond between these bases should be broken down so that it will get free. So the hydrogen bond between the bases should be broken down. It will break down. Then helix get opens and the synthesis of 2 new strand will takes place. So the result is 2 identical DNA double helix with 1 new strand and 1 old strand. This is the result of DNA replication. But I had said this in 4 steps, but it is not a simple task. It is so complex that it only takes place in presence of certain proteins and enzymes. So there are requirement of certain proteins or enzymes for this process. So this mechanism will be discussed in the part 2. So we are moving to discuss about the enzymes and proteins that are involved in the DNA replication. So there are different proteins and enzymes involved in the process. For example, helicases, then single-stranded binding proteins, topoisomerase, DNA polymerase, primase, ligase, etc. So DNA have to be copied to the daughter cell very quickly with less errors, so that there should be such efficient mechanism for the DNA replication. So we are going to discuss about the mentioned enzymes. Okay. First enzyme that come into this process is helicase. And one other enzyme is DNA topoisomerase that we will be discussed later. This will identify where to unwind the DNA and start unwinding of DNA. Then only the helicase can bind in that place. So what is the role of helicase? For replication to take place, the DNA should unwind the double helical structure. So helicase helps to break the hydrogen bond that holds the complementary bases of the DNA. So after unwinding the double helical structure by topoisomerase, then our helicase enzyme breaks the hydrogen bond between them, breaks the hydrogen. You can see in the picture, helicase is breaking the hydrogen bonds between the bases. Okay, So between the complementary bases, they are breaking the hydrogen bonds. So what is the role of helicase in DNA polymerase? Helicase helps in breaking down of hydrogen bonds between the complementary bases. Okay, everybody understand? Okay, we are moving to next enzyme, that is single stranded DNA binding proteins, SSB. This single stranded DNA binding proteins are very much important in DNA replication because these complementary bases have a tendency to base pair each other. So keeping it as such, that is keeping the DNA in single stranded form, certain proteins will help in this process. That is, this single stranded DNA will bound to the unwanted region, that is in the single stranded DNA. As the name suggests, this protein can only bind to the single stranded DNA, not to the DNA double helix. The SSB cannot bind to the DNA double helix. Not that point. This single stranded DNA binding proteins cannot bind to the DNA double helix. It can only bind to the single stranded DNA, so that when helicase breaks down the hydrogen bond, the single stranded proteins will come and bind to the single stranded DNA. Single stranded DNA, so that the bases cannot again base pair. Okay, it cannot form a hydrogen bond between the complementary bases. Everybody understand? So this single stranded protein binds to the newly separate. So this single stranded protein binds to the newly separated individual strands and keeping the strands separated. That means it doesn't allow these two strands to make hydrogen bonds between the complementary base. Everybody understand this point about SSB proteins? Yes, we are moving to next enzyme, the topoisomerase. We have already discussed about this in the first enzyme. That means these topoisomerases are enzymes that participate in overwinding or underwinding of DNA. That means when you open the DNA strand, if this is your double helix, this is your double helix. If you open this DNA strand, so you are opening the DNA strand, what will happen? Coiling of this region will take place. That is, this region will highly become coiled. That is, I have a problem with my touchpad, so that it will coming like this. Okay. So this is your replication fork. So your DNA is open. Then what happens to the DNA if you open from one side? It will highly coil. It will coil, highly coil. So like this. So what will happen? Tension will arise. The DNA can break. If we are doing so, the DNA will break out. So that for avoiding this tension, this topoisomerase will help. This topoisomerase do what? This topoisomerase will help in overwinding or underwinding of DNA. Okay. During transcription and DNA replication, the DNA needs to be unwound in order for the transcription or replication machinery to gain access to the DNA so it can be copied or replicate. So this topoisomerase have role in DNA replication as well as in transcription. So they are important in unmounting the DNA, and also it helps in overwinding and underwinding DNA. Topoisomerase have several roles. It have endonuclease activity. It has ligase activity. This topoisomerase, when this coil will form, supercoils will form, when supercoil will be formed in the DNA, this topoisomerase make a cut. It will make a cut and release the tension. After that, it join the DNA. So it have endonuclease activity as well as it have ligase activity. And also it have two other function, main function, that is it will unbound the DNA and it will help in overwinding or underwinding the DNA. For doing overwinding or underwinding of DNA, they use two activities, that is endonuclease activity and ligase activity. Okay, next we are moving to DNA polymerase. Okay, the main important, crucial, essential enzyme in the DNA replication mechanism, the crucial enzyme, the enzyme that synthesizes DNA. It will add nucleotides to the newly synthesizing DNA. This is one of the important enzyme in DNA replication. So the enzymes play an essential role in DNA replication. Okay. So DNA polymerase, we can say that DNA polymerases are the heart of the human DNA metabolism. Not only human, it also in each of the organism, the DNA polymerase will play a role, important role, a crucial role in DNA synthesis. Okay, so this DNA polymerase catalyze the DNA synthesis in template dependent fashion. Okay, template dependent fashion. This is your replication fork. So your DNA polymerase will add nucleotides. One thing is that the DNA polymerase have a drawback. It cannot initiate DNA replication. Always remember that. Please note this point. DNA polymerase cannot initiate DNA replication. It need a primer. When primer will binds to the template strand, the DNA polymerase will add nucleotides to the 3' end of the newly synthesizing DNA. And always remember that the new strand will be synthesized in the 5' to 3' direction. So DNA polymerase incorporate the deoxyribonucleoside monophosphate in 5' to 3' direction. Okay. So this is the important DNA polymerase. This is one of the important enzyme, DNA polymerase, that is very important in synthesizing DNA. Okay. So DNA polymerases cannot initiate DNA synthesis. They are not able to start DNA synthesis. They are fully dependent on a presence of free 3' OH group. This will be provided by our primer. Okay. So when primer will bind to the template strand, after that the DNA polymerase get a 3' free 3' OH group. A free 3' OH group. The DNA polymerase get a free 3' OH group. So that our DNA polymerase will start to synthesize new DNA. Everybody get it? Okay. Then this reaction is energetically driven by the fact that incoming DNTP chemically reacts with the 3' of the primer to produce the corresponding DNMP and pyrophosphate, which is finally hydrolyzed into inorganic phosphate. That means there will be one phosphate group will be present in between two nucleotides, and two phosphate will be released in the form of organic pyrophosphate. Okay. So we can discuss about next point. The error rate in the formation of AT and GC pairs are in the range of 10 raised to 3 to 10 raised to 5 depending on the DNA polymerase. So DNA polymerase is highly efficient. You can see that only one mistake in 10 raised to minus 3 or 10 raised to minus 5 mistakes. That means only one mistake in this nucleotides. That means error rate is very low when compared to RNA polymerase in transcription. RNA polymerase, when compared to RNA polymerase, the error rate is higher than DNA replication. So what are the function of DNA replication? DNA replication helps in synthesizing new DNA strand. The accuracy of DNA synthesis reaction is further enhanced by 3'5' proofreading exonucleases. So proofreading, the DNA polymerase have one function, that is DNA synthesis, and other function is proofreading activity. Proofreading activity is 3' to 5' direction. So the DNA polymerase have a 3' to 5' exonuclease activity, proofreading activity. Mainly in prokaryotes, we can see three different types of DNA polymerases: DNA polymerase 1, DNA polymerase 2, and DNA polymerase 3. In three of this DNA polymerase 3 is the functional enzyme for DNA replication. DNA polymerase 3. Okay. Next, we are moving into sliding clamp. So this sliding clamp is one of the important enzyme. We can say that it is an accessory enzyme, and it is also important one because this enzyme will help the DNA polymerase to constantly bind to the template strand. It doesn't allow the DNA polymerase to detach from the template strand. So a DNA clamp or sliding clamp, it's also known as beta clamp, and homologue of this sliding clamp in human is pCNA. So the DNA clamp is a protein complex. It's a protein complex that serves as a processivity promoting factor in DNA replication. So you can see that the DNA polymerase 3 holoenzyme is given in this figure. You can see that this red color thing is sliding clamp or beta clamp. This is the core enzyme. Core enzyme consists of alpha subunit, theta subunit, epsilon subunit, and gamma complex consists of gamma unit, then delta dash delta, then psi unit and chi unit. These are the holoenzyme of DNA polymerase, and this DNA clamp, this will help to bind the DNA polymerase constantly on the template strand. That is, this critical component of the DNA polymerase 3 holoenzyme binds to the DNA polymerase and prevents this enzyme from dissociating from the template strand. It doesn't allow the DNA polymerase to detach from the DNA. So this is your replication fork and your DNA is here. So here is the clamp loader. It will binds to the DNA as well as the DNA polymerase and doesn't allow the DNA polymerase to detach from the DNA will not allow to detach. So it will not allow to dissociate from the DNA template. This is the important role of sliding clamp. So we have discussed almost all proteins and enzymes that are involving in DNA replication. And one more thing I have to say is, when adding the nucleotide bases, you can see the beautiful animation here. When you add nucleotides to the newly growing strands of the DNA, here energy is required. Energy is required. How, from where this energy they get, from where they get the energy? So when this nucleotide comes here, it has 3 phosphate molecules, and this cleavage will provide energy, and only 1 phosphate is added in between every nucleotide. So this is 1 nucleotide, only 1 phosphate group, another nucleotide base, then again 1 phosphate group. 2 phosphate groups are cleaved and in the form of organic phosphate, and this will give energy for this reaction. So like ATP and other NTPs like GTP, TTP, CTP, that is nucleoside triphosphates, are high energy molecules. Important point, not this point. They are high energy molecules. They can serve both as source of DNA nucleotide as well as source of energy. So this nucleoside triphosphate can both act as source of DNA nucleotides as well as source of energy to drive the polymerization reaction. So when the bond between the phosphate is broken, the energy is released, used to form the phosphodiester bond between the incoming nucleotide and growing chain. So this bond between the nucleotide is known as phosphodiester bond. The bond between the complementary bases are hydrogen bonds. A bond between 2 nucleotide bases are phosphodiester bond. Note this point. Okay. So this is all about basics of DNA replication. Then we will see DNA replication mechanism in detail in next video. The main steps like initiation, elongation and termination, everything will be discussed in detail. So if you are preparing for CSIR, ICAR or DBT exams, you will get every points regarding the DNA replication. So it will be uploaded in the part 2. So please you see the part 2 video also for understanding the DNA replication. So if you like my video, please give me a thumbs up and please subscribe my channel. Also click on the bell icon so that you will get upcoming notification. Thank you for watching my video.